Welcome back. Now we are going to begin our discussion of Chapter 4. It is here that we will first start talking about what proteins look like. Up to this point, I have used the language of three-dimensional structure to describe the overall shapes of proteins, but here we are going to really get into what the details of that description look like. So when I say three-dimensional structure, what is it that I mean? Am I referring to just the shape? Am I referring to certain specific features of a protein? Here I'm going to dive deep into what that detail, what that characterization actually means. Before we move any further, I want to try to provide some perspective here. Up to this point, prior to Chapter 4, we have only talked about amino acids. So we have only talked about the building blocks of proteins. Proteins, in contrast, are much longer polymers of these amino acids. So the protein is the product of many condensation reactions where you have taken individual amino acids at different combinations and covalently attached them. So now, when we talk about a protein, what we have to really remember is that proteins are much larger than amino acids are. The figure that I'm showing you here is actually the protein chymotrypsin shown on the same scale as the amino acid glycine. So what I want you to see here is that chymotrypsin, a protein, is much larger than glycine. Now, part of that is directly relevant here. And what I mean by that is the protein, because it is a polymer of amino acids, it also has many more covalent bonds than individual amino acids do. So why does this matter? Well, this matters because this leads to the concept of conformational flexibility. And what I mean by that is each covalent bond is free to rotate. So if you have two atoms that are connected by a covalent bond, they are free to rotate about that covalent bond. Assuming that we are talking about mostly single bonds, free rotation can occur about the bond, which leads to different conformational possibilities. The spatial arrangement of atoms in a protein or any part of a protein is referred to as conformation. So a conformation for a protein is sort of an overall description of a collection of different rotational states about the many covalent bonds that exist in a protein. Now, just to remind you, in Chapter 3, I told you the difference between configuration and conformation, but here I want to remind you once again. To go from one conformation to another involves a rotational change. To go from one configuration to another, you have to break a excuse me, I'm not dying, I promise. To go from one configuration to the next, you have to break a covalent bond and change stereochemistry. So to go from D-glycine to L-glycine involves breaking of covalent bonds, but to change conformations does not. So the possible conformations of a protein or protein segment, this is a smaller part of a protein, really include any structural state that it can achieve without breaking covalent bonds. So because proteins do have many covalent bonds present, you can imagine the conformational possibilities are quite broad. We can sample many conformational states. A change in conformation could occur, just for example, by rotation about single bonds. Of the many conformations that are theoretically possible in a protein containing hundreds of single bonds, usually one, sometimes more, but usually one conformation predominates under biological conditions. The need for multiple conformational states that are stable reflects the changes that must take place in proteins as they bind to other molecules, such as substrates or other proteins, or even catalyze reactions. The conformations existing under a given set of conditions are usually the ones that are thermodynamically the most stable. That is, they have the lowest Gibbs free energy values. Proteins in any of their functional folded conformations are what we refer to as the native proteins. For those of you that take me for this class or any other class, usually I try to provide some historical perspective. I want you to understand generally when we knew some of the things that we now know, and I want you to also see that science as a whole is driven by humans. So what this, as you'll see later in the course, what this means is that, sometimes people do things with not the best motivations. Sometimes they are driven by, self-ambition or, you know, things that are not perhaps the most ethical concerns. So my point in saying this is that I want you to sort of get a feel for the humanity of science. Now, here, there's a lot less controversy. It's just sort of a serendipitous series of events. But we're going to start in the early 1900s. So prior to 1934, we knew very little about what proteins looked like. We knew to some extent what they were made of. We knew that there were some carbon, some nitrogen, oxygen, some other things. But it was largely assumed that proteins were just these amorphous blobs. They kind of lacked structure, at least that was the assumption. This was until 1934 when J.D. Bernal and Dorothy Crowfoot Hodgkin showed the first diffraction pattern for a protein. Now, I'll tell you what that means in a moment. Before I get there, I want to sort of point your attention, for those that are interested, in Dorothy Hodgkin. She is a pioneer of X-ray crystallography in the biological world, and she was responsible for solving structures of many biologically relevant molecules, and in a time when it was very difficult for women to enter science in the first place. So there are biographies written on her for those of you that are interested. Later, I will tell you about some other examples of women in science, not today, but in later chapters. But so I like to sort of highlight people as we go. Now, okay, she oversaw the generation of the first diffraction pattern for a protein crystal. So what do I mean by this? Well, if you look down here, you'll see this is what I'm referring to. This is a diffraction pattern. Now, let me tell you how we got here. Okay, so using a method that is called X-ray crystallography, what you do is basically generate a crystal of your molecule. In this case, we're talking about a crystal, a protein crystal. This is no different than, say, the salt that you use to season your food. There, your salt crystal is just a crystalline lattice of sodium atoms, chloride atoms, and they pack together in a repeating array. So the same thing is true here for protein crystals. Basically, you have molecules of protein that are packed together, much like bricks in a wall. They are packed in an ordered arrangement adjacent to other protein molecules. So what you're going to do is take your protein crystal, you're going to expose it to X-ray radiation, and the X-rays that are entering the crystal are going to be scattered out on the other side. So what you're going to do is you're going to place a piece of X-ray film, or in today's methods, a detector of some sort, on the other side of that crystal that is diffracting X-rays. So what you're seeing here are the scattered X-rays. Now, if your protein, or really your crystal, is ordered as a whole, what you should see is, due to constructive interference, these discrete spots. Okay, so these spots tell you where molecules are in your crystal. Now, I'm not trying to make you experts on X-ray crystallography, especially not at this level. But what I want you to understand is that in an X-ray diffraction pattern, seeing discrete spots like you see here can only be possible if your molecule is ordered. So in 1934, when Bernal and Hodgkin showed a diffraction pattern for the protein pepsin, that told us for the very first time that pepsin, a protein, was not a random colloidal structure. It wasn't just this, random ball of spaghetti. It was instead an ordered array of atoms. Seeing that diffraction pattern with discrete spots can only be possible if your molecule is ordered. Okay, so as early as 1934, we knew that proteins were not just this random mess of atoms, but instead was, they were some sort of ordered array. Now, at the time, in 1934, you couldn't really go much further than this, at least not for a protein. But as time went on, it became more technically possible to take these data and actually use them to construct an atomic model for a protein, or really any biological molecule. Without going too deep, what you can actually do is take the distance between the spots and the symmetry. So you'll see here there is a hexagonal sort of shape here. You can take that information and use that to actually construct an atomic resolution model for your protein. Okay, so I've actually been showing you myoglobin. And this is interesting because in 1939, we had a diffraction pattern for myoglobin. Now, this at the time was not sufficient to actually generate that atomic model. So the first atomic resolution model for myoglobin, in fact, this is the first atomic resolution structure for any protein, was reported in 1958. So that is quite a few years ahead of 1939 here. So that was for sperm whale myoglobin. Now, why sperm whale? This is kind of it seems kind of random. Well, this is an interesting story, in fact. So the researchers that were involved in trying to determine the structure of myoglobin, they basically had tried all kinds of different source materials. So myoglobin itself is an oxygen transport protein that is present in all mammals. We need myoglobin to transport oxygen to muscle tissue. So they had tried different kinds of mammalian tissues, or basically what they're doing is they're obtaining meat from different sources and purifying, isolating myoglobin from that source material. So one of the ideas that came up was, okay, what mammals are more likely to have higher concentrations of myoglobin? Well, the answer that they came up with was diving mammals. So think whales or dolphins, or basically mammals that have to stay underwater for long periods of time. They would need to have more myoglobin so that oxygen could still be delivered in the absence of oxygen. So when you're underwater, you can't breathe so easily. I think we can all appreciate that. Now, this is where the serendipity comes into play. So these researchers, or at least some part of them, maybe one guy was on vacation in South America, and he came across some sperm whale meat. And he decided, I'm going to take that home. I'm going to purchase some sperm whale meat. And remember, this was in the 1950s, so I'm guessing that customs was not asking the questions they would be now. But I would have to ask, as a customs agent, why are you bringing home 15 pounds of sperm whale meat? That seems odd. But at the time, it was possible, and they did this. And so they were able to obtain sufficient amounts of myoglobin from that South American sperm whale meat that was obtained via methods I don't really know all the details to, but they were able to obtain enough myoglobin to crystallize it and solve the first structure. So here is that structure. Now, what I want you to notice here is that we still don't quite have atomic resolution. You're basically looking at a bunch of blobs. But what you should see is that there is some order. Now, one other bit of historical reference to point out here is that only 5 years earlier, so actually in 1953, Watson and Crick had just reported a model for double-stranded DNA. So double-stranded DNA, as you'll see later in this course, it's just a helix. It's a fairly simple structure. So when the structure from myoglobin was reported here, protein chemists and really biochemists more broadly did not immediately believe this because they thought it was too complex. They didn't see an apparent regularity in the structure. With DNA, there is a clear regularity. Everything is ordered, and it's a pretty simple structure. Those perspectives were really what guided the apprehension towards this structure. Like when people saw this, they thought, that's a little bit more complex than DNA, and I am skeptical for that reason. I expect it to be, a little bit more closely related to DNA. But as it turns out, this was just human bias that sort of got in the way. This was in fact correct, and as you'll see going forward, protein structures do tend to be more complicated than nucleic acid structures. Now, this 1959, or I should say 58 structure, it was the first protein structure that we ever had. For that reason, myoglobin is very well studied to this day. However, it took another 10 years to reach a resolution shown here sufficient to actually describe the protein in atomic resolution. Now, what I want to point out here is, you're looking at this thing thinking, okay, that's a structure, there's probably some meaning, but at this point, I expect that you're going to be viewing this very much through the eyes that my mother-in-law views this. My mother-in-law looks at structures like this. She's seen me bring home textbooks, and she says, "Oh, that's really nice party confetti you have there. I don't know what it means, but that's nice party confetti." And that's what I'm assuming of you at this point. I want you to just basically trust my word when I say, "Oh yeah, this actually means something." So this helix here, that has some meaning. I don't expect you to be an expert on this yet. You will, but not yet. But what I do want you to see at this point is that there's something in the middle here. This is where the atomic resolution allowed for insight. So over here in this low resolution structure, you can't really see stuff, any individual atoms in the middle. But here you see, based on color, we can tell you exactly what atom is present and exactly where it is. That is what I mean when I say atomic resolution. Okay, so now having said all of that, I know this slide seems like it's taking forever and ever, but having said all that, what I want you to see here is that these initial studies of myoglobin have opened the door to the study of a vast array of proteins that are quite variable with regards to complexity. Some are as simple as myoglobin. Yes, I said simple. Others are far more complex in structure. But the interesting thing is that there is, in fact, a pattern. There is a remarkable degree of structural regularity that you see. And as we move forward in our discussion of protein structure, you're going to see how this sort of plays out. Now we turn our attention to the hierarchy of protein structure. What you're going to see here is that there are, in essence, 4 layers of structure that build upon one another. And all of this is made possible by non-covalent interactions that are provided by the functional groups inherent to the individual amino acids. And so I'm going to tell you sort of what I mean by this as we go on. So the lowest layer of structure is simply that of the amino acid sequence. This is shown here in part A. This is what we refer to as primary structure. So here, all this is the sequence of amino acids laid out from N-terminus all the way down to C-terminus. So that gives you a very specific look at the order of amino acids that would come in the polypeptide chain and what functional groups are present therein. Okay, so the next layer of structure occurs when you start seeing non-covalent interactions occurring between amino acids that are close in primary sequence. So that leads to the formation of what we refer to as secondary structure. Here, you're seeing one type of secondary structure. This is the helix, and we will talk about the alpha helix later in this chapter. Now, Secondary structure, again, it forms via interactions between amino acids that are close in primary sequence. Usually this is going to involve not side chain interactions, but rather interactions between the backbone atoms. So if you remember when we talked about the peptide bond, you have a repeating pattern. You have a C alpha carbon, carbonyl carbon, nitrogen that is going to be there leading to peptide bond formation. So those are the atoms that are going to really drive secondary structure. Now, again, secondary structure forms as a consequence of non-covalent interactions between atoms that are close in primary sequence. Now, the next layer of structure is tertiary structure. This is the level of structure that forms upon formation of non-covalent interactions between amino acids that are distant in primary sequence. Okay, so usually what this looks like is secondary structural elements interacting with other secondary structural elements to give you tertiary structure. And if you have a single polypeptide chain, this is probably it. This is as far as you're going to go. This is your three-dimensional structure. Now, to restate, secondary structure is structure that forms as a consequence of interactions between amino acids that are close in primary structure. Tertiary structure is structure that forms due to interactions between amino acids that are more distant in primary structure. Okay, so that really sums up all of the structural levels, the hierarchical structural descriptions for a single polypeptide chain. But often you're going to find protein complexes that form via interactions between distinctly different and unique polypeptide chains. In part D here, this is your first look at something like that. This is actually the protein hemoglobin, or the protein complex hemoglobin. There are 4 different polypeptide chains, and they are numbered beta 1, beta 2, alpha 1, alpha 2 here. So 4 distinct chains come together and form a larger protein complex. This is what we refer to as quaternary structure. So once again, primary structure, that's just your amino acid sequence. Secondary structure is structure that forms via local non-covalent interactions, interactions between amino acids close in primary sequence. Tertiary structure forms by interactions that are occurring between amino acids that are distant in primary structure. And quaternary structure forms as a consequence of interactions between distinctly different polypeptide chains. So if you have a single polypeptide chain, then tertiary structure really gives you the description of three-dimensional structure, and in fact could be also a substitute. It could be a description of quaternary structure. So if you have a single polypeptide chain, tertiary structure may well be equivalent to quaternary structure. Okay. If you have multiple polypeptide chains in your complex, then quaternary structure just describes the interaction that occurs between all of those chains. Now, when we talk about these in shorthand, you'll see these 2 degree sign, 3 degree sign, 4 degree sign, or you'll have a 1 for primary structure, but 1 is primary structure, 2 is secondary structure, 3 is tertiary structure, and 4 is your quaternary structure. So try to remember this. We're going to talk about this a lot in different ways, but I will be repetitive. This is something that you really need to know, this concept of protein structure hierarchy. This is an important one. Biochemists of the 1950s were really caught off guard by the complexity of the myoglobin protein structure. It was a lot more complex than they expected it to be. So before we move on, I want to take a moment to sort of address why protein structure is so complex. To get at the punchline, protein structure is simply a thermodynamic compromise. What we are doing is finding that structure that minimizes the overall free energy. So this does boil back down to thermodynamics. Now, if you think about what a polypeptide is, it is a polymer of amino acids where each amino acid has a side chain functional group. It has a carbonyl group that is found in the backbone. It has a nitrogen that is involved in peptide bond formation. So there are many functional groups within a polypeptide that ultimately are capable of both non-covalent and covalent interactions that give you the final three-dimensional structure. We just talked about this in the context of protein structural hierarchy. So non-covalent interactions really are the primary driver of protein structure, and these non-covalent interactions ultimately lead to a lowered Gibbs free energy. Okay, so remember, protein structure, it is complex, but ultimately it is being driven by the need to find that lowest energy Gibbs free energy value, that low energy state that would correspond to the final structured state. Now, before I proceed, let me give you a couple of definitions. When we talked about amino acids, we talked about configurations. There are D amino acids, there are L amino acids. Just as a reminder, a configuration, to go from one configuration to another, requires a change in stereochemistry. It requires a change in covalent linkage. So you have to break a covalent bond to go from one configuration to another. Conformations, on the other hand, simply require rotational alterations. So if we think about this in the context of a polypeptide, there are many covalent bonds, many of them, if we think about this in terms of peptide bonds, there are many peptide bonds that are capable of rotation to give you a large number of different conformational possibilities. So I'm going to show you an example of this. And remember, we're looking at conformational differences, not configurational differences. So what you're going to see here is a simulation where this unstructured polypeptide, so no real structure here, samples different conformations, and these conformations really lead to the formation of non-covalent interactions between functional groups that are found in this polypeptide. And this is going to be tracked by a conformational landscape. So you'll see this sometimes. It's sometimes referred to as a protein folding funnel, free energy landscape, or conformational landscape. But all it is a representation of different conformations. So if you look like up in red here, these are largely unstructured conformations. You'll see why this makes sense in a moment. But as you move down this funnel, so from top to bottom is Gibbs free energy. So those unstructured conformations, basically something looking like what you see here. These are high energy states, and ultimately they're going to sort of walk down this funnel, and in order to do that, they're going to have to form non-covalent interactions that ultimately lead down the funnel to the lowest energy state, which is shown in purple down here. You're going to see this lowest energy state is going to be structured. There is going to be three-dimensional structure. So the formation of three-dimensional structure is ultimately going to lead to a minimized Gibbs free energy, that lowest energy state, the most thermodynamically favorable state. So now I'm going to show you the video. Okay, so unstructured right now, we're sampling different states up here. Now we find our way down and we hit some structure, and ultimately we're going to end up in the bottom of the well. Let me jump back to the end there. We're going to end up at the bottom of the well where we have structure. So this three-dimensional structure is formed through first the formation of secondary structure, and that secondary structure formation then leads to the formation of your final three-dimensional structure that corresponds to your lowest energy thermodynamic state. We've been focusing on structure that forms as a consequence of non-covalent interactions, but we really, before we go any further, need to talk about the peptide bond itself, which is a covalent interaction. So we talked about this. The peptide bond is that characteristic linkage that connects amino acids in a polypeptide or a protein. It is an amide linkage. Remember, you have the carbonyl carbon of one amino acid that undergoes condensation with the alpha amino group of an adjacent amino acid to connect two amino acids via this amide linkage. This is the peptide bond. Now, the peptide bond itself has a rigid planar structure, and this is the consequence of resonance interactions that give the peptide bond approximately 40% double bond character. So here you've got two different resonance states where these pi electrons of the carbonyl group can exist over here, or they can hop over here to give you the carbon-nitrogen double bond. So there is approximately 40% double bond character. Now, this knowledge, this comes from X-ray diffraction studies of crystals of amino acids and also of simple dipeptides and tripeptides. And these studies showed that the peptide CN bond, this carbonyl carbon nitrogen bond, is somewhat shorter than the carbon nitrogen bond in a simple amine, and that the atoms associated with the peptide bond are also coplanar. So this indicated a resonance or partial sharing of two pairs of electrons between the carbonyl oxygen and the amide nitrogen. The oxygen has a partial negative charge, and the hydrogen bonded to the nitrogen also has a net partial positive charge, which also sets up a small electric dipole moment too. So altogether, you've got a double or a partial double bond that is occurring right about here, which keeps these atoms coplanar. However, there are two other covalent bonds that we have to think about too. So we have this C-alpha carbonyl carbon covalent bond and the C-alpha nitrogen bond on the next C-alpha position. So together, these three, 1, 2, and 3 covalent bonds are all approximately coplanar and form this basically coplanar set of atoms where you have one plane per peptide bond. So if you think about this in the context of a larger polymer, a larger polypeptide or protein, every peptide bond basically can be thought of as a plane. So here if we look at a larger polypeptide, you can see that for every peptide bond, the 6 atoms of the peptide group lie in a single plane, such that the oxygen atom of your carbonyl is always going to be trans to the hydrogen atom on the nitrogen, the amide nitrogen. So here we can conclude that the peptide carbon-nitrogen bond, because of its double bond character, cannot rotate freely. So this peptide bond in a given plane, so here you have one, here you have another, here you have another. So these peptide bonds cannot rotate freely. However, rotation is permitted about the C-alpha nitrogen, so C-alpha nitrogen, and the C-alpha carbonyl carbon bonds. And you basically get free rotation allowed at these points. The backbone of a polypeptide chain can thus be pictured as a series of rigid planes with consecutive planes sharing a common point of rotation at each C-alpha carbon. The rigid peptide bonds limit the range of conformations that are possible for a polypeptide chain. Now, before I move on, one other thing to note here is that the peptide bonds assume the trans conformation in which successive C-alpha carbons are going to be on opposite sides of the peptide bonds joining them. And you should sort of take note of where your side chain R groups are. So for this plane, C-alpha, or your side chain functional group, the R group, is pointed upward. On the next position, it's pointed down. On the next one, it's pointed up. And this is just a steric compromise. Otherwise, you would have steric clashes between the side chain functional groups. Now, the cis conformation, where your R groups are pointing in the same direction, is about 8 kilojoules per mole less stable than the trans conformation because of the steric interference between neighboring side chains. In some cases, the steric interference is going to be reduced in peptide bonds associated with proline residues, but for the most part, you're going to find the trans conformation is going to be more stable than the cis conformation. Okay, so now let's talk about how we actually define conformations. So first, conformations of peptides are going to be described by torsion or dihedral angles. I'll tell you what this means momentarily, but peptide conformation is going to be defined by three dihedral angles called phi, psi, and omega. These reflect rotation about each of the three repeating bonds in the peptide backbone. So first, phi represents rotation about the C alpha nitrogen, C alpha amide nitrogen bond here that I just told you about. Psi represents rotation about the C alpha carbonyl carbon bond. And I told you that there are three dihedral angles. There's also omega. Omega is going to refer to the peptide bond where there isn't a whole lot of rotation. Now, a dihedral angle is the angle at the intersection of two planes. For peptides, the planes are defined by bond vectors in the peptide backbone. Two successive bond vectors describe a plane. Three successive bond vectors describe two planes. And the angle between these two planes is what we measure to actually describe peptide conformation. Now, I'm using this term backbone, main chain, but I haven't really defined what this means. So when we talk about the backbone or main chain of a protein, these are going to involve the atoms of the backbone, really, of a protein. So the carbonyl oxygen, the carbonyl carbon, the amide nitrogen, the C alpha carbon. These are all backbone or main chain atoms. So going forward, when I refer to the backbone or main chain atoms, this is what I'm talking about. I'm not talking about side chain functional groups. I'm talking about the backbone, I'm trying not to use the word in the definition, but the atoms that are repeating across the entire polymer. Okay, so now we've talked about conformations and sort of I've defined the important bond angles for you, but let me tell you how to use them. Now let's look at an example. So I just told you that a dihedral angle is the angle at the intersection of two planes. Phi and psi both represent dihedral angles occurring at the intersection of two planes. So psi, remember, is the covalent bond that connects C alpha and carbonyl carbon. It is also a dihedral angle that represents the angle at the intersection of these two planes formed by adjacent peptide bonds. Okay, so looking at psi as an example, both psi and phi angles are going to be 180 degrees when the polypeptide chain is fully extended. Okay, so here I'm showing you the fully extended case where your amide nitrogens on adjacent C alpha carbons are trans to one another. This is the 180 degree definition, just by convention. The value of phi or psi is going to increase in the clockwise direction, and you'll see how this works in a moment. Now, if we just look down the C alpha carbon, just take this, rotate 90 degrees, and view it down the C alpha carbon, you'll get a sense for what I'm talking about. So for simplicity, I'm going to start not in this position, but rather in the cis position. So if you can imagine, just bear with me for a moment. I take this nitrogen, rotate about this bond so that it's pointed downward so that the amide nitrogens are cis to one another. This would be my 0 degree position. Okay, so when the nitrogens, the amide nitrogens are cis to one another, they are defined as having a dihedral angle of 0. Now if I rotate 60 degrees, okay, 60 degrees, I am increasing in value. So now at this position, I'm positive 60 degrees, move another 60 degrees, 120, until I hit 180, which corresponds to this original state I showed you where the amide nitrogens are trans to one another. This is by definition the 180 degree position. And then as I continue in the clockwise direction, now my values, I'm still changing in 60 degree increments, but I now have a negative sign. So I'm at minus 120 here, minus 60, back to 0. Okay, I'm going to do this in the least awkward way possible, or at least what I think is the least awkward way possible. I wanted to give you a different view of these dihedral angles. So here I have just a little model that illustrates sort of how you might view this. So here you have C alpha carbon. I don't have an R group on here, so you'll see this is just empty. This is where the R group would be. Hydrogen on the C alpha carbon. Alpha amino group here shown in blue. Carbonyl carbon connected to the C alpha carbon, but carbonyl carbon here in black. Carbonyl oxygen in red. And here, right here actually is my peptide bond. I've got my carbonyl carbon connected to my next amide nitrogen, so this is my peptide bond. Now, if we're going to look at this through the lens of psi, if you remember, I just told you that when my two amide nitrogens are trans to one another, that would be what I define as my 180-degree position. So if I do this kind of like this, you should see now that these are trans to one another. Hopefully you can see that. If you look down the C alpha carbon, okay, this is awkward to do in class, much more awkward to do by video, but you should see that they are fully extended in this way. Now, this would be 180 degrees. If they're pointed cis to one another, like that, I'll put that down so you can see. If they're pointed cis to one another, same direction, this is the 0 position. So this is 0 degrees. And if I begin rotating 60 degrees, so here you should see you're kind of out of plane. If you're looking down, let me do this in the least awkward way possible, looking down the C alpha carbon, they are in the same plane. The amide nitrogens are sterically going to bump into one another potentially. If I start rotating, so 60 degrees, approximately 60 degrees, now they are offset. So this is my positive 60-degree psi dihedral angle. Another 60 degrees gives me the 120-degree value. Now I'm in the fully extended, doing this in, again, the least awkward way possible. Now I'm fully extended. If I keep on going, this would be my minus 120, minus 60, and now I am back to 0. So hopefully that was not too painful, but I just want you to get a sense of the spatial relationships between these dihedral angles. Hopefully you noticed just now that there are certain dihedral angles, in particular the 0-degree position for psi, where you may have steric clash. At that 0-degree position, my amide nitrogens, if you look over here for reference, remember they were pointed cis to one another and potentially bump into each other, which presents a steric issue. There are other combinations, other phi or psi dihedral angles, where rotation about the C alpha nitrogen and C alpha carbonyl carbon bonds form certain combinations of phi and psi that cause the amide hydrogen, the carbonyl oxygen, or substituents on C alpha positions to collide, which is what you're seeing here. So here you have a steric collision between the carbonyl oxygen and the amide hydrogen. So what this should tell you is that in principle, phi and psi can have any value between minus 180 degrees and positive 180 degrees, but many values are actually going to be prohibited by steric interference between atoms that are contained within the polypeptide backbone and the amino acid side chains. So not all dihedral angles are going to be physically possible for reasons of steric interference. So how do we assess sterically allowed versus sterically forbidden phi-psi combinations? Well, if you remember, every amino acid, or rather every peptide bond in a polypeptide is going to have a distinct phi and psi value that corresponds to the dihedral angles for each of those characteristic bonds. Okay, so what we can do is use a tool that is referred to as the Ramachandran plot, named after a man named G.N. Ramachandran. This is a topology plot that puts phi on the y-axis and psi on the x-axis. So phi versus psi. And if you remember, every polypeptide is going to have one phi and one psi per peptide bond within. So each of these dots represents basically an XY coordinate that corresponds to a single peptide bond. So for every peptide bond, you have a phi and psi. If you treat them as an XY coordinate, then you can plot per peptide bond each of those phi-psi combinations. Now what you should notice here is that if we break this up into 4 quadrants, some quadrants are much more populated than other quadrants. So quadrant 2, if we number this 1, 2, 3, and 4, quadrant 1 and 4 are not as well populated. So why is that? Well, that's because these are quadrants where basically you have nearly all, you have a lot of sterically forbidden values of phi and psi. So these are values that would lead to steric clash between atoms in the polypeptide. So quadrant 4, really you hardly ever see anything because all of these phi-psi combinations are sterically forbidden. You get far fewer phi-psi combinations that show up in quadrant 1 for the same reason. There is a little island here highlighted in purple where you do get some allowed values, but as you'll see going forward, this corresponds to a left-handed helix, which doesn't really show up in nature a whole lot. So you're not going to see a whole lot of that. But quadrant 2 and 3, you get much more densely populated XY coordinates. It's because these are the regions where you do have sterically allowed conformations that are accessible. So this should sort of get to you the heart of the Ramachandran plot. It is a very useful tool for assessing sterically allowed versus sterically forbidden values of phi and psi. Most of the diagram actually represents forbidden conformations, but you'll see these densely populated areas correspond to distinct secondary structures. I'm not going to go into in great detail what they mean just yet, but you'll see, just for example, there's a helical structure, a right-handed helix that shows up that would correspond to this region of quadrant 3. And there are these things called beta sheets that show up that would correspond to phi-psi values that show up in quadrant 2. So we're not quite to the discussion of secondary structure yet, but what I want you to see is that the Ramachandran plot here allows you a way to assess sterically forbidden versus sterically allowed. Now this is actually useful, very, very useful in fact, if you are building a protein model based on X-ray diffraction data, because remember, any model that we ever look at is a model. It is a model that represents the most likely positions of atoms, but it is still a model nonetheless. So it is a model based on experimental data. Now, the Ramachandran plot is a tool that allows you to assess model quality. If your model has a Ramachandran plot that has a whole lot of data points in the 4th quadrant, there's probably something wrong with your model. You probably should work, and look for these steric clashes that would likely not be allowed in real life. So the Ramachandran plot presents a quality control tool that can be used when constructing atomic resolution protein models. Okay, so this marks the end of part 1 of chapter 4. And here we spent a lot of time talking about the peptide bond. Its coplanar arrangement of atoms, its partial double bond character. But what we didn't really focus on were the functional groups within. Going forward, when we're about to enter into a discussion of secondary structure, you're going to see that secondary structure, also tertiary structure, also quaternary structure, is structure that is really dependent upon the functional groups that are contained
within the polypeptide. So going forward, you're going to see more of a focus on non-covalent interactions. But before we can get there, we had to sort of lay the foundation. We had to talk about what the peptide bond is, how it behaves. So just remember that this is a foundation that we're building from.